Now on to translation. Before we actually talk about the process of translation, let's talk about the composition of ribosomes, starting with the prokaryotic ribosome. Let's start with the two subunits together to make the full ribosome. Prokaryotic ribosomes are 70S. They have a small subunit and a large subunit. When they are together, they are 70S. The large subunit by itself is 50S. The small subunit by itself is 30S. And no, you didn't do your addition wrong. When the large subunit comes together with the small subunit, some of the projections get covered up, so it sediments at a faster rate than the two separately, if you were to add them together, would do. The small subunit is made up of 16S rRNA, ribosomal RNA, and 21 polypeptides. They all come together to make a functional 30S subunit. Remember the 16S ribosomal RNA. It was studying that allowed us to find out that archaea are significantly different than bacteria. For the large subunit, we have 5S rRNA, 23S rRNA, and 34 polypeptides. Now, looking at the eukaryotic ribosome, it's larger and it's got a different shape. Especially if you look at the small subunit, you can see that it's got a different shape than the prokaryotic ribosome. Now remember that archaea have 70S ribosomes. This is the shape of the bacterial ribosome, the one that we're most familiar with. Archaea have this size, but remember they have more of this shape. So the two subunits of the eukaryotic ribosome, coming back to eukaryotes, we have a 60S large subunit and we have a 40S small subunit. When we're looking at relatedness between eukaryotic organisms, we take a look at the 18S rRNA, which is the equivalent of the 16S. So why do I go into all of this? One, so that you know what the parts are when we talk about the process of translation. Also, later on when we talk about taxonomy in more depth, then you'll be familiar when we talk about 18S rRNA and 16S rRNA. Now, the reason why we take a look at the genes for this ribosomal RNA is because it can't change very much. If it changes too terribly much, it affects the enzymatic ability of the ribosome. Basically, the ribosome is unable to construct proteins and the organism dies. We know that any mutations that happen have to happen in areas that don't affect the active site, and we also know that mutations are going to happen at a fairly consistent rate in those other areas that don't affect the active site. Now let's talk about how ribosomes, either eukaryotic or prokaryotic, take the information in the mRNA and turn it into a protein, a process which we call translation. Like transcription, we have initiation, elongation, and termination. Let's start with initiation. The small ribosomal subunit binds to the mRNA so that the start codon is in what will be the P site, P for peptidal, or think of it as P for protein. Then the first tRNA with the first amino acid comes in. We call this the initiator tRNA, and the anticodon, or the RNA bases that match the start codon, comes in. So the anticodon matches up with the start codon, and the tRNA, which is loaded with its amino acid, comes in. This takes energy. During all of translation, you're going to see that we're using GTP rather than ATP. This is a way that the cell uses to regulate which processes are which. So ATP gets used in catabolic reactions, it gets used in quite a few anabolic reactions, but GTP is used in translation. We have our small subunit here. The anticodon on the tRNA has hydrogen bonded to the codon on the mRNA, and the large subunit clamps over the top of this. so that we have the tRNA in the peptidyl site. We have the A site, which I like to think of as the addition site, and then we have the E for exit site. That is the end of initiation. Let's move on to elongation. The first thing that happens is a new tRNA with a amino acid has to come in. Comes into the A site, the anticodon has to match the codon on the mRNA. So now we have two tRNAs with two amino acids attached. What happens next is the large subunit creates a peptide bond between the amino acid on the first tRNA with the amino acid that is on the second tRNA. So this amino acid gets clipped off of the tRNA, attached to the next amino acid, which in this case is phenylalanine. So the methionine in prokaryotes is formal methionine, in eukaryotes it's just methionine. We have a dipeptide of the formal methionine and the phenylalanine. The ribosome moves over one codon on the mRNA. Think of it as a little train on some train tracks. So now the tRNA that has the growing peptide is now in the P site, and the empty tRNA is now in the E site, and the A site is freed up. Now this movement of the ribosome also requires GTP, which we're not showing here. So coming up here, the empty tRNA leaves to go get another F-methionine. Then another loaded tRNA comes in and its anticodon matches the codon in the A site. Once again, the peptide is moved from the tRNA in the P site, and it is covalently bonded through a dehydration reaction to the amino acid on the new tRNA. So now we've got a tripeptide. This keeps going on throughout elongation. We have loaded tRNAs that come in, the empty tRNA comes out, the growing peptide is moved to the amino acid on the new tRNA. This is elongation. For termination, when the ribosome gets to the end of the mRNA, to the end of the coded part of the mRNA, it will reach what is called a stop codon. The stop codon does not code for any tRNAs. No anticodons match the stop codon. What happens is a termination protein will come into that site, and it will cause the two subunits of the ribosome to separate, and the peptide will come off of the last tRNA, and the tRNA will come off of the mRNA, and that is termination. Now I'm going to put in an animation that shows what we've been talking about. Hopefully it'll reinforce what you're learning.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Genes can't be left on all the time. If we were to leave genes on all the time, unless they're important products that are needed all the time within the cell, then that would be very expensive and the cell would go bankrupt, so to speak. It would have a light bill it couldn't pay for. So there are ways of controlling genetic expression so that genes are turned on and turned off so that their gene products are available only when the cell needs them. So let's take a look at the lac operon, which is a set of genes in E. coli that controls the metabolism of lactose. That's where the lac in the lac operon comes from. So the lac operon is composed of the promoter, what is called the operator, and all of the genes needed to metabolize lactose on the line. Now, upstream from the promoter for the lac operon is the regulatory gene that controls the lac operon. That's all together nice and neat. So what happens is the regulatory gene is transcribed and results in a repressor protein being formed. This repressor protein binds to the operator so that when the RNA polymerase binds to the promoter, it can't move forward and transcribe the structural genes. So in this way, E. coli does not produce the genes needed for the catabolism of lactose except for when they're needed. Well, how does the cell know when they're needed? When we get a lot of the byproduct of lactose, allolactose, when the concentration gets high enough, it'll start binding to the repressor. Now, the repressor doesn't have the spot where it can bind to the operator. So the repressor is no longer there. RNA polymerase can bind to the promoter, go through and transcribe the structural genes of the lac operon. When that concentration drops, when the cell has used up the lactose in the environment, the allolactose will fall out of the repressor will rebind and will go back to the initial state. We call this an inducible operon because it's induced by what the genes are needed for. The tryptophan operon is kind of the opposite. It's still set up as an operon. We have the structural genes for making the amino acid tryptophan all together. You have an operator, and you have a promoter, and you have a regulatory gene over here. But E. coli makes tryptophan continually unless tryptophan is in the environment. So the repressor isn't there unless tryptophan is in the environment. So tryptophan comes in, binds to the repressor, makes it so that it can fit onto the operator and block transcription of the genes needed to make tryptophan. Well, why would the cell do that? Well, if it's in the environment, it's cheaper to import it than to make it yourself. So we call this a repressible operon. In other words, it's on most of the time. Unless there's tryptophan in the environment, then it gets turned off. So it's kind of set up the opposite of how the lac operon is set up. There are other ways to regulate genetic expression. One is through microRNAs. These are small stretches of single-stranded RNA that form with proteins to form an RNA silencing complex. They bind to mRNAs that have sequences that match the microRNA. And they can either snip through that mRNA or they can just keep the ribosome from clamping onto it. Small interfering RNA is similar to microRNA, but it's double-stranded. still forms with proteins to form a RNA silencing complex, and it does pretty much the same thing. Well, why would a cell want to do that? Well, suppose the lac operon has been turned on and the ribosomes are busy translating the mRNAs into proteins needed for lactose metabolism, but now we've run out of lactose. So this is a way to silence mRNAs that have already been made, whereas the repressor of the operator is a way to keep the mRNAs from being made. Riboswitches are another way of silencing mRNAs from keeping them from being translated once they've been transcribed. Now, a lovely example from the book is Yersinia pestis. Yersinia pestis causes bubonic plague. Nasty disease, caused death and destruction for many, many years, and it's still around. Well, Yersinia pestis produces a virulence factor, which causes the plague in humans. Well, they're constantly transcribing this because you want to have the mRNAs ready for when they're exposed to the humans so that they can take advantage of that environment as soon as they get there. But you don't want to be translating it until it's needed. So what happens is the mRNA folds into a complex pattern that the ribosome cannot attach to and translate. The riboswitch is in this fold at room temperature. Once the cell gets to body temperature, in other words, when the cell leaves as an invertebrate flea and gets into the nice warm bloodstream of a human, then that mRNA unfolds. The mRNA can now be translated into the virulence factor. Isn't that clever? Well, that's it for this topic. Here are some tables that I hope you'll find useful. And here are reminders of what you need to learn from this topic.